engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
Discoveries.
Advances.
Questions. Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists podcast, the programme that brings you the biggest breakthroughs and talks to the major movers and shakers in the worlds of science, technology and medicine with me, Chris Smith. Coming up, a new drug that blocks malaria in a whole new way and turns future mosquito bites into vaccine boosters. Also, England is close to eliminating hepatitis C. How's it been done, and can the rest of the world follow suit? And a designer vest to protect astronauts from space radiation gets tested on the Artemis missions. Scientists in Australia have developed a new anti-malarial drug that targets the stage at which the Plasmodium parasite moves out of liver cells, where it grows initially, and into the bloodstream. Now, crucially, blocking that process not only prevents the disease becoming symptomatic and stops it spreading to other people, it also turns out that holding up the parasite like this leads to a very powerful immune response against it, which can block subsequent infections, potentially indefinitely. It also means that any other mosquito bites that occur in the meantime act almost like vaccine boosters, bolstering the existing immune response and reinforcing the protection. It is a whole new way to halt malaria and to make people immune at the same time, and it's the brainchild of Melbourne's Walter and Eliza Hall Institute researcher Justin Boddy.
we currently have control measures for malaria. There's three lines of defense, really. Insecticide-treated bed nets, anti-malarial drugs, and vaccines. The problem is that mosquitoes have developed resistance to insecticides, and that the two approved vaccines that we have, which were indeed major breakthroughs, are imperfect because they don't provide long-term broad protective immunity. So we need additional vaccines. And parasites are constantly developing resistance to the antimalarials that we developed, and we have some amazing antimalarial drugs. So we really have quite a problem on our hands that as we continue to fight malaria, it continues to fight back.
And what are you doing to try and take that fight to the parasite?
We started by embarking upon a drug development program where we're trying to develop drugs that are active not just against the parasite when it infects red blood cells, but when it's moving through its life cycle between mosquitoes, the human liver and the human blood, and then back to mosquitoes again. And so when you're developing new antimalarial drugs, what you'd really like to do is have activity at multiple stages of the parasite's life cycle, because it provides you a number of different options. You may have heard of the saying, prevention is better than cure. And so what we've embarked upon here is the idea that we can actually use antimalarial drugs not only to sort of treat malaria, but also to prevent malaria before it can take hold. And in doing so, we've unveiled that this drug can elicit a long-lasting immune response in preclinical models that we didn't really expect.
Can you explain a bit more then, Justin, how this actually works in terms of how a drug that can block a parasite can also bolster an immune response like this?
Sure. It all comes down to the way that malaria is spread. So mosquitoes bite people and they inject these little parasite forms that are called sporozoites. And sporozoites are very small, and they can come out of the proboscis of a mosquito, and they go into the skin and then go into the liver, and they'll actually invade liver cells and grow inside them. And those people have no idea that they're infected. It's effectively silent. There's no symptoms, there's no awareness that the person is infected. So this lasts for a week to 10 days, and then the parasite comes out of the liver and infects red blood cells. And that then initiates malaria, the disease, and all of the pathophysiology associated with malaria. So what this drug does is it actually prevents the malaria parasite from transitioning out of the liver and into the blood. So quite remarkably, the drug permits these sporozoites to invade the liver, and it permits these liver stage parasites to grow and replicate in quite large numbers exponentially, but it kills them right at the end of their liver stage, just before they can infect red blood cells. And it happens to be the late liver stage is a bit of an immunological sweet spot for priming both CD8 T cells, but also antibody responses that together can provide multiple lines of defense.
And presumably then, if a mosquito comes along and re-challenges a person with malaria again while they're on this agent, then they're going to get a big immune response to that liver stage, which is permitted to proceed. But then it never translates into clinical disease because the parasite's blocked again at that stage. So they get all the benefits of the immune priming, none of the disbenefit of the disease.
You've hit the nail on the head, Chris. That's why we're so excited about it. And I think what this unveils is the possibility that, unlike current vaccines, which are really important, where you need sufficiently high input numbers of sporozoites to elicit a strong protective immune response, this particular drugging approach allows a small sporozoite input approach to still prime immune education. And as you've rightly identified, then each subsequent bite by the mosquito is going to seed the same opportunity for immune education, but it's in a boosting environment. And so we know that our immune systems really like to be boosted. That helps them to maintain immunity, but also extend broad immunity. So malaria can be a problem because there's different variants of it, much like COVID. And so if there's areas where there's more than one variant that would be existing in a population, this approach could also provide protection against these different variants through this boosting approach each time a mosquito bites.
Tell us about the evidence you've got that this is going to work, though, because it sounds like a wonderful approach. It sounds like an amazing theory if it works, but what's the evidence that this really can deliver?
Yeah, it's a really important question. And so I think we need to obviously be reserved in this, but very carefully present the evidence that we have. So the evidence is twofold. Our study is in preclinical models using different strains of mice. And so there's malaria that infects mice. And mice can be quite predictive. Sometimes they're not predictive, but mice can be predictive for malaria vaccination. And in our mouse models, what we've observed is that just a single encounter of sporozoites that are given to the animal in the presence of this new drug that we're developing with our pharmaceutical partners, that is enough to not just protect them from disease, but to protect those animals for the rest of their life. And so we're having near lifelong immunity from a small encounter. And we tested this across a range of different mouse strains that have in themselves different immune arms. So in some mice, you get strong antibody responses, but not such strong T cell responses. And in other mouse strains, the converse is true. And so what we're able to show is that we do get both strong and long-lasting antibody responses, but also long, what we call tissue-resident memory T cells that are primed and boosted, and they'll hang around, particularly in the liver, waiting to recognize infected liver cells through a variety of different parasite antigens.
Does this require the individual to remain on the drug, though? So is this drug only effective while you keep taking it, like every other anti-malarial we've got at the moment?
So that is where we would love to investigate this further in humans. So with mice, in the future, the drug was no longer present. The drug has a half-life of about 2.5 hours in mice. And when we challenged those mice even 2 years later with mosquito bites, they were still completely protected against getting malaria. So there's this long-term protection that this particular mechanism is priming. And so what we'd like to understand is whether or not that's true in humans, because if so, that would be potentially quite game-changing for how you might use a drug to prevent malaria in vulnerable communities.
Brilliant, isn't it? Just embody there at the Walter and Eliza Hall Institute in Melbourne, Australia. That research just out in the journal Science. NHS England has said the UK is leading the world in efforts to eliminate hepatitis C. This bloodborne virus infection, which was only discovered in recent decades, affects hundreds of millions of people around the world, and it's a major risk factor for liver cirrhosis and liver cancer. But the development over the last decade of a suite of new drugs that specifically target the virus has turned what was a chronic disease with a poor prognosis into something we can routinely cure and potentially even eradicate globally, because us humans are the only host. UK deaths from the infection are already down by a third, with England one of just a handful of nations to have already surpassed the World Health Organization benchmark towards eliminating the agent from our shores. Will Gelson is a consultant hepatologist at Addenbrooke's Hospital in Cambridge, and he kindly joined us from his holiday in Normandy, where he's hopefully not punishing his own liver too severely with Calvados that Normandy is known for, to bring us up to speed.
Hepatitis C is a bloodborne virus which infects the liver and can cause cirrhosis and liver failure as well as liver cancer in some of those who are infected with it.
What fraction of the population, both in the UK but also globally, are thought to have this?
The fraction is around 0.5% globally, so it's a relatively common disease. It varies geographically quite significantly, though.
So, point five, I'm just trying to work that out. So, that must mean there's a couple of 100 million people around the world who are affected. And this causes cirrhosis. Is that all it does, or does it have other implications?
Well, in about 20% of people infected with the virus, it will cause cirrhosis, and cirrhosis is a risk factor for liver failure and liver cancer.
And so, obviously, it's great news that we now have ways to treat it. The treatments that we've got, do they work for everybody?
Almost everybody. So there's a first round of treatment which involves 8 to 12 weeks of a tablet that is taken once a day, or 3 tablets that's taken once a day, that will completely cure the virus in 95% of those who take the medication. For those who don't clear the virus, so that 5%, 1 in 20, there's a second round of treatment that itself is over 90% effective. So there's a very, very tiny proportion of patients in whom we cannot clear the virus and cure them. But of course, that's a very small proportion.
Does this mean then that we're really within reach of being able to eradicate this? With that level of intervention, that quality of intervention, we could potentially kick Hep C into touch.
We could potentially, as you know, very few infectious diseases are actually eradicated, and we're trying to head towards elimination at the moment. So eliminating it as a public health threat, but certainly with medications as efficacious as those, we should be able to achieve huge improvements in the treatment of hepatitis C for populations at large.
Because I gather the UK is something of a flagship example here. We're ahead of the curve in the initiative of trying to get rid of it from our population.
That's right. So NHS England have set up a very effective programme of treatment. They established good value for money for the treatments, and a programme of testing and treating those with the virus. The World Health Organization have a target of 2030, and we've certainly had a significant impact on that already in England. So we've treated over 80% of the known cases, which is one of the targets. We're already at 36% reduction in deaths with a target of 65%. So I don't know quite when we'll hit it, but hopefully by the 2030 target, we will have hit the majority, if not all, of the metrics that the World Health Organization have given.
How are we finding cases? I mean, as in, when you're a hepatologist, you will see people and manage the treatment, but they've got to be found in the first place and referred to you. So what sorts of initiatives are out there to go and find people in the population? Because this is very much a silent infection, isn't it? People often don't know they've got this, which means it's much harder to then track them down in the first place and help them.
That's correct. So there are lots of strategies. One of them recently has been accident and emergency testing. So in high prevalence areas, ED testing is going on. There are initiatives in some general practices. In prisons, there are high intensity testing and treatment initiatives. In drug and alcohol services, similar programs. And each area of the country has been given a pot of cash to test and treat in a way that is most beneficial for their population.
And lastly, if we do nail this one, what sort of a difference to your practice is this going to make? As in, at the moment, how much of a headache for a hepatologist is hepatitis C, and therefore how much is getting rid of it going to save the health service?
This has had a huge impact on my practice already. When I started as a consultant hepatologist, we had a good proportion of liver transplants that were undertaken for liver failure secondary to hepatitis C. And they were very difficult to manage after transplantation because the virus invariably came back, and the treatments that we had would actually cause rejection of liver transplants. And now not only can we treat people after liver transplantation, but they generally don't get to liver transplant in the first place. The other area that has made a massive difference already is liver cancer. So it's now relatively rare, unfortunately not completely stamped out, but relatively rare to get a cancer from hepatitis C. So it's made a massive difference in our practice already. So it's a moving feast, really, Chris, but certainly it's just changed hugely over the course of the last 10, 15 years.
A phenomenal success, isn't it? Hepatologist Will Gelson there from Addenbrooke's Hospital in Cambridge on the successful fight to help eliminate Hep C. And as Will points out, if you are in the UK, you can obtain a home test kit to test your Hep C status. Details are at hepctest.nhs.uk if you want to check it out.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
This is the Naked Scientists podcast with me, Chris Smith. Now, we're halfway through today's programme, so I wanted to mention our other Q&A show, which is called Ask the Naked Scientists. And if you want to hear it, find out more, or even contribute to it, then you can do that by heading over to nakedscientist.com/ask, or look up Ask the Naked Scientists wherever you get your podcast. No question is a stupid question, and it's always great fun. Still to come, how a new toothpaste uses frozen smoke to relieve the pain caused by sensitive teeth. But first, researchers have created a special vest to protect astronauts from space radiation. Tests using the uncrewed Artemis 1 mission showed that the AstroRad vest can significantly reduce the radiation astronauts receive during a major solar particle event or a solar storm. Oren Milstein is the CEO and co-founder of STEMRAD, which is the company behind it. He and his colleagues have used two torso models that mimic human tissue in the Orion spacecraft, and they found that their garment would have lowered radiation exposure to levels seen in two past solar particle events. It's also flown on the International Space Station, with real humans trying it. I've been speaking to him.
Radiation, be it on Earth or in space, is daunting a problem that needs overcoming, especially radiation that is very penetrating. So those thin lead aprons that we're all familiar with from the doctors, they don't do much when it comes to penetrating radiation such as gamma radiation or space radiation. So STEMRAD has tackled this problem by use of very sophisticated wearable radiation protection.
What sorts of doses do astronauts get at the moment?
Yes, so astronauts venturing into deep space will receive doses much higher than those that are in low Earth orbit on the International Space Station or in the past on the Space Shuttle, where they received doses that are probably about 40 times what you would receive here on Earth on a daily basis. And that's not pleasant, but going into deep space, that could catapult to about 500 times more than what you'd be receiving here on Earth. So think about it that within the span of a week or two, you can potentially receive a dose that's higher than what you receive here on Earth for your whole lifetime.
We tend to talk in terms of radiation exposure in terms of what we regard as a safe exposure, short-term and lifetime exposure. So if we take, say, a journey to Mars, that will be about 9 months at shortest. What do we anticipate the dose would be in terms of lifetimes worth of radiation exposures that an astronaut going to Mars would get, for example, then?
Going to Mars, in terms of lifetimes, it would be in the thousands fold of what you'd receive here on Earth. So it would take you 1,000 years here on Earth to get what you would get on the path to Mars and back. So yeah, it's significantly higher, and that translates to significantly higher likelihood of getting cancer down the road. And that's what we're trying to tackle here by mitigating that amount by as much as we reasonably can.
And what's your approach? What is the technology? How does it work?
Yeah, so shielding the whole spacecraft is not feasible, just be too heavy. And weight is really the bottleneck in getting to these destinations. So rather than trying to shield the whole spacecraft, we resorted to shielding the actual human body. That's a lot more mass efficient. So you're able to achieve pronounced radiation attenuation by use of a physical mass, and specifically in space, a mass that is hydrogen rich, such as polymers, polyethylene, etc. So we take those materials and we adhere them to the body in proximity to the most susceptible organs in the body, such as the bone marrow and the gastrointestinal tract. And in the case of women, the breasts are very sensitive. So we place them in proximity to those very sensitive organs, while the thickness of the shielding really reflects the underlying susceptibility of each tissue being protected.
So effectively, you end up looking a bit like a superhero. You wear a suit that has extra defenses in your most vulnerable bits, and it's using
Yeah,
chemistry to soak up any incoming radiation, because you're putting something in the way of the incoming radiation, and that just happens to be
exactly.
these polymers. Um, is this light and practical though? Because one of the things astronauts are going to say is, I need to move around, contort my body into all kinds of spaces, and space is at a premium in space. So is it a
Right.
practical solution?
Yeah, so to really observe how practical the solution is, we first of all sent it to the International Space Station, the ISS, and it spent a couple of years there being worn by 5 different crew members to get exactly that feedback as to how practical the solution is. So that was a preliminary prototype, and the purpose of that was to get that feedback. And indeed, we got a lot of feedback based on which we were able to improve the ergonomics of the product. So as far as comfort and ergonomics, the product today is at the point where it could be worn for days on end and even slept in. And that's very important because solar particle events can last up to 1 week. So it's not something you need only for a few minutes, but rather for several days sometimes. So that was one side of the equation that we successfully solved. The other side is really the mass limitations at launch. So mass is, as I mentioned earlier in this conversation, is very challenging to move from here to lunar orbit and especially to Mars, where you're going to see a very harsh limit on what could be sent, to the point where people are even rationed water and food in going to these remote destinations. So to answer that, we came up with the solution that we thought was the most mass efficient, and that's the Astroret at 26 kilograms. But since then, and given the success of this study that's being published today in Science Advances, we were able to further reduce the mass towards 16 kilograms, knowing where we're overprotecting and where we're underprotecting based on this mission that we just did on Artemis 1. We were able to reduce the mass to the point of about 16 kilograms without compromising the protection too much.
What's it actually made of, though? Because I know you said you're using chemistry, hydrogen, etc., to get in the path of the radiation, the cosmic radiation, but some of these things are very penetrating. Some of them have got enormous energies. So how are you able to soak them up? And to what extent can you soak up the radiation? How much does the dose drop by?
The vest can block up to 60% of the radiation coming from a very violent solar flare, such as the one that took place in 1972 between Apollo 16 and 17. So that's very, very important. And then, as far as the material that we're using, so we're really using hydrogen-rich polymers. In the case of Artemis 1, we used HDPE, simply high-density polyethylene. Sounds very simple, but it's actually one of the best materials in the world when it comes to blocking particulate radiation, such as exists the protons coming from the sun in the case of a solar flare.
Oren Milstein, who's the CEO at STEMRAD. And if you want to see for yourself what Oren's new vests look like, he has sent us a picture that accompanies the transcript of his interview. You can have a look at nakedscientist.com. Just click on the podcast link. You'll see this show listed there and the interviews that are in it, including this one. Tooth sensitivity affects around half of UK adults, causing sharp pain that can put people off cold drinks, ice cream, or even brushing properly. Most sensitive toothpaste can only mask the symptoms, either by numbing the nerves in our teeth or slowly blocking the microscopic tubules that get exposed in worn-down teeth, and that takes days of repeated use. Now, an NHS surgeon has taken a different approach, borrowing a material from aerospace and bone graft science. Aerogel is an ultralight porous solid used occasionally by NASA and nicknamed frozen smoke. NASA use it to capture micrometeors in space. Upon contact with saliva, the new material releases sequestered calcium and phosphorus ions to seal up the tubules within the teeth within just minutes to rebuild a protective mineral layer on the tooth and reduce the sensitivity. It's the first newly patented approach to tooth sensitivity in almost 3 decades.
So my name is Niall Kent. I'm an oral and maxillofacial surgeon who works in London and the co-founder of an oral care brand called Ozone.
This deals with the problem of sensitive teeth, doesn't it? So what is that and who's got it?
Sensitive teeth affects approximately 50% of the population, and it's the short, sharp pain, which is often and commonly caused by cold triggers, so cold drinks, but it can also be caused by hot things, tea, coffee, and also very sweet things.
And if you look at the tooth, what's actually going on to produce that unpleasant effect?
Yeah, so your tooth has got a number of layers. It's got the blood vessel and nerve inside, and this is what's removed when you have a root canal treatment. And then you've got dentine, and then you've got enamel. So it's like 3 layers. When the dentine becomes exposed, these little tubules that run inside the dentine become exposed, and the cold triggers or the hot triggers or the sweet triggers cause a sensitization of the nerve inside the tooth, which gives a sensation of pain. The dentine becomes exposed when either the enamel is worn away or you get recession of the gum.
And what is it? Because what's in the tubules gets cold or hot, and that transmits that to the interior nerve in the tooth.
Yeah, so you've got fluid that exists inside these tubules, and it's expansion or contraction of this fluid, which then causes the sort of sensation of pain within the nerve of the tooth. So cold things cause contraction and hot things cause expansion.
And how have you sought to tackle the problem?
What I've done is developed a novel material called Aerograft, which is a bioactive aerogel, and it's a material which acts to block these tubules and prevent these triggers from interfering with the fluid and the nerve.
How does it work?
So it works by releasing calcium and phosphate, which are bound within a sort of aerogel structure, which once it's put into your mouth and mixed with the water in your saliva, it releases the calcium, releases the phosphate, and precipitates hydroxyapatite, which is the mineral that bones and teeth are made of, onto the tooth surface, which blocks the tubules and acts like a sort of varnish to prevent sensitivity.
There's a lot of calcium and phosphate already in toothpaste. If you look at the back of the tube, you know this as a Max Fax person. Yep. It's loaded with the stuff. So why don't I just go and get a tube of existing toothpaste and smear that around my gums and get the same effect?
So the issue with the calcium phosphate that's in toothpaste at the moment is the fact that it's crystalline. What that means is it's got a very ordered structure, but it's also not very soluble. The difference between that and the Aerograft is that the calcium phosphate is amorphous. Okay, what that means is it's highly soluble and can be released into saliva to precipitate or to crystallize and form that mineral. So what's in the existing toothpaste is not going to be released. It's not going to form a layer on the tooth surface, whereas the Aerograft will do that.
And where does the aerogel aspect come in? Why do you need that at all?
So that acts as a kind of carrier for the calcium and phosphate. It's a bit like a kind of fishnet. So it's like a chain of silicon atoms, which are then have the calcium and phosphate bonded to that. And they keep the calcium and phosphate amorphous, so it's not crystalline, so it's highly soluble. And they also act as a sort of building block once it's inside your mouth. And that mineral forms as well as on the tooth surface, but also in the particle. So when the particle goes inside the tubule, you get the precipitation of the mineral on the tubule and on the tooth surface.
Presumably that takes a little while for that effect to be achieved and therefore for the relief to come. So how quickly upon beginning to use this would I, if I had sensitive teeth, begin to see a benefit?
So if you look at so we've looked at it in the lab and it takes less than 30 minutes to do that reaction. Okay. Whereas the existing materials take around 22 hours. So we've dramatically decreased the time it takes in the lab to do that. How that translates to people actually using it. So we've recently done a clinical trial involving 88 participants where 89% of them had a reduction in sensitivity on the first time of using the product.
And is that what it is? It's just used as a toothpaste. So instead of using your normal brand, you would use your product and get the effect, or do you need to use this as an adjunct to existing tooth cleaning regimes?
The best way to use it is to not rinse your mouth out after using it and just spit out any excess and leave it on the tooth surface. But we've also invented an oral gel product, which is kind of like a concentrated product, which is used for times other than brushing, where you typically get sensitivity. So you can use it around teeth whitening, or you can also use it, you're going outside, you're going to the beach, you can have a cocktail, and you can put it on before that to prevent sensitivity before those kind of like peak periods.
How does a maxillofacial surgeon end up playing around with this stuff? How did you stumble upon how to do this?
One day I was meeting my brother for coffee, who's an artist at the Royal Academy of Art, and he'd gone to a lecture on aerogels, and what he wanted me to do was to make him a sculpture out of aerogel, because they're quite kind of interesting materials to look at. I sort of agreed to do this for him, and when I started looking at the chemistry of aerogels, I realized that they would be an excellent kind of medium for holding these calcium and phosphate ions. Originally developed it for a bone graft because, well, various reasons. One, the low density. The hypothesis was that low density would mean that the material would resorb and remodel very fast. And then once I started looking in the lab at how quickly this reaction was happening, I sort of realized this would be an excellent material for solving sensitivity.
And it's completely safe. There are no health problems. It's biocompatible. You can put this in the body, no problem.
Yeah, I mean, the only thing that it's made of is silicon, calcium, and phosphate, which all exist inside the body. We've registered the ingredient as an INCA ingredient, so it's gone through quite rigorous testing and assessment. And then we've also, as I said, performed an 88-person clinical trial, which has shown no irritation.
It really is an example of science meets art, this, isn't it? What does your brother say, having given you the idea?
He's he's
The
still
word you're
very
looking for
proud.
is shareholder, is it?
Yeah. He still wants to make the sculpture. I kind of quickly dropped in the sculpture. No, he's sort of very proud because it's also all of these things. They're not just like one person doing it. They're kind of lots of people in their contribution, and he has quite a big contribution to this.
Noel Kent, who's an oral maxillofacial consultant and also co-founder of Ozone, a new aerogel-based way to treat sensitive teeth. Well, that's it for today. I did promise you last week, though, that I was going to report back on my taste test of Savour's artificially made butter, which we talked about in a previous programme on Foods of the Future. Do check that out. It was a great listen last week, even if I say so myself. The sample did eventually arrive, and I tried it on a bit of baguette. It looked and tasted just like a pale butter. In fact, I wouldn't have known it wasn't butter if I hadn't been told. So synthetic butter it might be. Tasty it definitely was too. Join us on Tuesday. We're going to be examining wounds and wound healing and why people with diabetes are at so much risk. And that programme, brought to you in partnership with UK Research and Innovation, will be exploring the cost of wounds to healthcare providers and how science might have some new solutions in the works. Do please, meanwhile, pen us a review on whatever podcasting platform that you use to get the show. Those rankings really help our visibility, and that really matters, so thanks in advance for that. And if you appreciate what we do for you each week, do please consider making a donation to support the show. You can do that at nakedscientist.com/donate. And thank you very much from the bottom of our hearts to everyone who does chip in each week to keep the show on the road. I'm Chris Smith, and from all of us here at The Naked Scientist, thanks for listening, and until next time, goodbye.